So getting right into ingestive behavior, this is a bit of a continuation of what we were talking with just this past class on gustation and olfaction. And so in some ways it fits with a nice theme, and in many ways, some of the things we're going to be talking about are a little bit unique. When we talk about ingestive behaviors, one of the really important things that we need to understand is that this is a system that's designed for homeostasis. And we have a few different things that we monitor as a part of this system, and those are called system variables. So this is some sort of a property that we're trying to control. And for the case that we're looking at now, the things we're trying to control in our body would be hunger or thirst. And all of these systems, and this is why I've encouraged you to think about the brain and every interaction that it has with the body as a system, all of these systems have what we call a set point. And this is really an optimal value. So for all of us, we can imagine what this optimal value might look like, especially for something like hunger. There's a point where clearly we feel pretty good, we've eaten enough food, and you haven't overeaten, you're not bloated, you haven't taken in too much, but you're also not hungry and especially not starving. And all of these systems have some set of detectors, right? We need something to be able to measure how far we are from the set point. Am I too far above or below, or am I just right? For all of these homeostatic systems, we are looking for that Goldilocks effect. We would love for us to always be just right, but that's rarely the case. And because it's rarely the case, we also need correctional mechanisms. We need some sort of indicator that we've deviated too far from a point of homeostasis and that we should start engaging in some kind of an action. Hey, you should stop eating food because you've eaten too much. Or, you should really start looking for food because you're starting to get pretty hungry. And of course, with these correctional mechanisms, we need to have a feedback system for them, where we finally tell the correctional mechanism that's been triggered that it's done its job and now it needs to shut down. A really good example of the cycle that we're talking about comes straight from your book. So I'm certain that as we head into the colder months of winter here, that all of you are starting to turn on some kind of heater in your house, whether it may be a local space heater or the entire HVAC system. And when we do this, what we're trying to do is control the air temperature. So this ends up being the system variable that we're interested in. And the way that we figure out, at least in the house, what the air temperature is that we use a detector. And the thermostat is actually that detector. It determines first what the temperature in the house is. And then on top of that, we set a value on that thermostat, which is what we want the air temperature to be. And so the detector is looking for the current temperature, and then we pick some kind of a setting that may change depending on the individual, but it's going to tell us where we would like this to be. And if there's a deviation between the detector and this set point, then what happens is that we trigger a correctional mechanism. And in the case of our household systems, the correctional mechanism is for the heat to turn on, whether it be, again, a space heater, an electric heater, these tend to have thermostats built into them, or the HVAC system of your house, which will turn on the furnace. So as this happens, what we end up creating as a part of this is, of course, we're creating heat, and that ends up being the negative feedback system. Now, although you could probably all follow this red arrow here, I'll draw my own arrow anyway, which is that this negative feedback is, of course, going to come and it's going to then change the air temperature, which is then going to change the reading on the detector, and we'll be able to reevaluate whether or not this deviates from our set point. So what we're trying to achieve in our body for systems like hunger and thirst is satiety. And satiety is really just the idea that we've had enough. You are satiated, you've hit your limit. And this means that the body has everything that it needs to be able to continue working and acting, and we don't need to seek out any more food or water. However, unlike the idea of the thermostat, where the heat in the room, maybe it takes a couple of minutes to hit the thermostat, but it's a pretty instantaneous feedback. And yet for things like thirst, we need to wait for all of the different cells in our body to really absorb and redistribute water that we take in, even though we might have a lot of water that we've just put into our stomach. And for hunger, the signals take even longer. So what we end up doing is we actually measure both that system variable, or in the case of thirst and hunger, sometimes many system variables that tell us whether or not we are still thirsty or hungry, but we also measure how much effort we're putting into these correctional mechanisms. And the idea here is that the brain can kind of make predictions based on all kinds of information about whether or not we are headed in the right direction for satiety, or whether we should continue to work to find food and water, or take in more food or more water. So let's talk just a little bit about what would happen with one of these two-part systems for correcting some sort of imbalance in our bodies. So if we have a system variable like body fluids that we're monitoring and the body loses too much water, then what's going to happen is that we're going to have a set of detectors that are going to let us know that we've fallen out of sync with where we would like that system variable to be, right? We're not at our set point any longer. And this is going to trigger some sort of correctional mechanism. In the case of thirst, this is pretty obvious. It would be drinking. And once we have had some water, then our stomach is going to fill up. And what's going to happen here is we're going to initiate kind of both of these different mechanisms simultaneously. So the first mechanism is going to be something like the satiety mechanism you see here, where we use information not necessarily that we're back at our set point, that our system variable is back where it needs to be, but instead what we're going to use is information from the stomach. So as the stomach fills up with water, then we have stretch receptors that say, you know what, the correctional mechanism that we've been using, where we've been putting water into the stomach, has triggered something saying the stomach is full, and this starts inhibition following this path of the correctional mechanism. We slow down our drinking or stop our drinking altogether. And of course, this is going to keep looping, but it could be the case if we're really thirsty that this loop will continue to repeat itself. Our stomach will empty a little bit, but the correctional mechanism will still be getting signals from the detector, and so we'll stay in this loop for a while if we're really, really thirsty. But at some point, we're actually going to hit true homeostasis, where our body has absorbed enough water to where its cells and its blood pressure and all of these other things are back to the states that they want to be in, and this eventually will reset our system variable. So what you can see here is that we use these two different systems simultaneously in order to keep ourselves on track. So then what happens in the case of thirst? What are the signals that start to trigger all of these changes in our behavior? So thirst, as you all think about it, is just this behavioral drive to seek and consume water. But the thing that you may not know is there are actually two driving forces behind your thirst. The first one is the amount of fluid that your cells have to use. So what we're trying to do here is to find a balance between the amount of salt and other solutes in the body and then the fluid that the cells have to perform all of their different functions. And the other is actually our blood volume. So we need to maintain a certain amount of intravascular fluid, or fluid in our vascular system or bloodstream, in order to have normal heart function along with other things. So the very first type of thirst that we looked at that involves the amount of fluid that's around our cells is something that we call osmometric thirst. And the way that we monitor this is that we have cells in the body that are responsible for doing this, and these cells happen to be sensitive to stretch. The amount of stretch changes depending on the ratio of water to salts or solutes. So if we have water that leaves the cell because we become dehydrated, then those stretch receptors relax and become less stretched out. And for these neurons, they actually signal that by an increase in their firing that says, hey, I'm thirsty. So in this particular case, their firing rate is going to go up. On the other hand, when we end up with a whole lot more water that enters the cell, then this is now going to take these same receptors and it's going to stretch them out, just like when your stomach gets kind of stretched and bloated when you've had a full meal. These cells stretch when there's more water inside of them, and they have more water than they do have these salts or solutes. And what happens there is that the firing rate of these neurons is then going to decrease. So depending on the ratio of water to salts and other solutes, then we're going to have a change in firing rate that represents just how thirsty we are. And you can imagine, of course, this is not completely black and white or up and down. It's a graded scale where we have greater firing rates with greater amounts of thirst and lesser firing rates with lesser amounts of thirst, but it becomes a full continuum. So these osmoreceptors in the brain are located in a part of the brain called the lamina terminalis, and there are actually two different structures that play a role in monitoring our osmotic thirst. The first is this OVLT, or the organum vasculosum of the lamina terminalis. Easier to remember is OVLT. And then we also have the subfornicular organ. So what's special about this lamina terminalis is, of course, it's a center for thirst, but another thing that makes it special compared to the rest of the brain is that it is located outside the blood-brain barrier. So remember, we talked about a few special places that have really direct interactions with the body, and this is one of them. We monitor thirst by having this special set of neurons outside the blood-brain barrier. So there's been one really cool thing that's come out of human fMRI studies, and it's actually surprising that they've been able to do some of the things that they do. Because in these fMRI studies, they've found that they can dissociate two elements of thirst. The first of these is an element that relates to our anterior cingulate cortex, and this area is involved in a lot of things. We typically think of it as a part of cortex that is involved in processing emotional states, and so this area seems to relate to the unpleasant feelings of thirst. And then simultaneously, we can see neurons in the organum vasculosum of the lamina terminalis. I'm not certain if your book was trying to point to that in this little area here, but it's actually here. So their arrows might be just a little bit off, but these seem to represent the amount of solutes or the amount of hydration that a person has. And the last thing is, remember, there's some ways that we can use fMRI where we can look at connectivity, and we can look at this both in structure and in function. And so what fMRI has been able to tell us is that these two structures are functionally connected. So the greater activation of this OVLT that we have, then the greater activation of sort of these unpleasant feelings we're going to have in the anterior cingulate cortex as well. The second kind of thirst you need to know about is called volumetric thirst, and this is that one that relates back to levels of blood plasma. So really volume of blood is what we're talking about here. And we can have changes in these for many different reasons, but the most common change is just evaporation of water through our skin and while we're breathing. We expend a lot of water all the time just by existing and doing what we do. But there are some other reasons that can cause this to happen more rapidly, and of course, one obvious one is loss of blood. But if we also have sickness behaviors like vomiting or diarrhea, these can also cause dehydration. And this is in some ways a protective mechanism because vomiting and diarrhea help us to get toxins out of our system, but if they have to work too hard to do this, then they can actually make almost any sickness worse. So if at any point you're getting sick, this is why we always push fluids and try and stay hydrated, because everything in your body is going to work a little bit worse, including your brain and mental capacities, when you are experiencing this volumetric thirst. So we detect changes in blood volume and problems with our blood volume via two different mechanisms. The first of these is through the kidney. And so the kidneys are involved in processing a lot of fluids through the body, and they have a really complex vasculature. And it turns out that they produce a hormone that our brain can read called angiotensin that actually acts as a neurotransmitter in the brain. And so if we change angiotensin, we can start to change our drinking behavior and also our salt appetite. And one of the reasons this tends to work is that our kidneys use the angiotensin to increase blood pressure. And so when the kidneys detect kind of a lack of blood volume, then they increase blood pressure so that we can keep all of our organs and other things functioning, and their job is to protect the body. Then the second set of information comes from the heart, from a set of receptors called baroreceptors, and these are sensitive to changes in blood volume, and they're actually the type of receptor that in our body monitors blood pressure. So these two in combination are sending signals out that become very important to the brain. The neurons that get this angiotensin signal are in this subfunicular organ, or the SFO. And we know this because we can manipulate the activity of the SFO. So if you take and inject angiotensin right into the subfunicular organ, then you can drive drinking behavior. And if you use blockers, you can actually do the opposite. You can inhibit drinking behavior. So this tells us that this is really heavily involved. It's a critical structure. It's necessary, not just sufficient, but necessary to drive drinking behavior. And then one really important thing, especially because we have these two different categories of thirst, is that we need to put both of them together. And so there's one structure in the brain called the median preoptic nucleus that has the job of integrating all this information. And so if you actually look here at the positioning of our OVLT and our SFO, then one thing that you're going to notice here is that both structures are pretty much positioned on either side of this third and really important structure, the median preoptic nucleus. So this median preoptic nucleus is going to get input from these subfunicular organ neurons that are processing the angiotensin signals, and then most of the osmoreceptors are located in this organum vasculosum of the lamina terminalis, and those are sending signals to the median preoptic nucleus about the osmometric thirst. And then we have a third input that we don't even see mapped here, which comes from the nucleus of the solitary tract. And this is a place we already saw in looking at our gustatory system. This is a place where a lot of signals come from our taste buds, but part of the reason that this signal passes through there is that 10th cranial nerve, the vagus nerve, passes through this nucleus of the solitary tract, and so it has a really important role in multiple aspects of homeostasis, keeping our body in check. And one of the signals that it gets are from these baroreceptors detecting blood pressure signals coming from the heart. So what we see happening, and the reason we know that we have this complete integration of all these signals, is if you cause damage to the median preoptic nucleus, then what we end up producing is a behavior we call adipsia, which is that animals will no longer drink. So you eliminate completely the need for thirst. And this shows that this area, again, is not just sufficient, but is in fact necessary for driving thirst behaviors. And here we go with part 2 of our lecture on ingestive behavior. We're going to be talking a little bit about hunger here, but as I keep saying, if you just listen to part 1, go take a walk, go take a break here. Ingestion of food. So what we're trying to do, and we saw a little bit of evidence of this in the introductory section of the gustatory lecture, is that we're trying to find a balance of all kinds of different nutrients that we need so that we can store them, we can use them long term, and convert them into fuel. And this is all part of one bigger process of homeostasis. When we talk about short-term energy storage, the body has a pretty simple process for doing this. So we have cells that are either in the liver or the muscles, and they can store energy for a few minutes, maybe even up to a few hours is kind of the time range that we think of for short-term energy storage. If you think about as long as you can run before you start to get hangry between meals, that's kind of the time frame that we're talking about. And what happens here is that the fuel that we use when we need to break down something to get short-term energy is called glycogen. And this process actually starts when we take in things that have glucose, which many of you know as one form of sugar. So it's a simple sugar. And what happens is that insulin from the liver is going to be released, and it can take this glucose and it can convert it into glycogen. And then when the body needs to take this glycogen and then use it for short-term energy, it's going to do the exact opposite, where it's going to take the glycogen and it's going to use something called glucagon, which is kind of this opposite for insulin in some ways, and it takes the glycogen and breaks it back down into glucose so that we can use this for short-term energy. So when we're trying to store things long-term, then we use our fat tissue. The thing that's important about the fat tissue is that there are actually two components to it. We have the triglycerides, and then we also have, within these complex molecules, a substance called glycerol. So what happens is that we have cells in the fat tissue that can take all the nutrients out of the blood from things that you've eaten, and they can turn them into triglycerides as we need them, or convert them back into fuel for the body. And the way that this works is that the fatty acids that come from the triglycerides, all of these lipid components, are the things that are used by the body. And then the liver takes the glycerol that's in the triglycerides, and it's going to convert this into glucose so that we can use it in the brain. Another thing that's really important in the way that our body signals ingestive behavior is what our feeding state is at the time. And this will seem pretty straightforward. If you're hungry, you're going to have different signals than if you're satiated or don't need any food. So when we're hungry or fasting, then what we have is that the pancreas stops secreting insulin, and it's going to start secreting this glucagon that we need to begin converting glycogen over to glucose. And for the short term, most of this glucose is going to be used by the brain. Remember when we talked in the methods class, we talked about the irradiated deoxyglucose that we use for PET scans and other things of that nature. And the reason we do this is because the brain is the biggest user of glucose or sugar in the entire body. It needs a lot of energy to run and maintain all of these very active processes that it has. As time goes on, and if we continue to fast longer and longer, this is the point where we begin to draw from our longer-term stores instead of just short-term stores. In these long-term stores, we're going to begin drawing from our fatty tissue, as we just discussed. We'll take from those long-term reserves. And then in the very, very long term, if the body hits the point where it can no longer keep up at all, using fat or using simple sugars that we've taken from moderately complex carbohydrates, we'll start taking proteins from the body. So we'll actually start eating away at our own muscle tissue and other things like that. And one of the things that should be pointed out here is the whole goal of this is the very last things to go, the very last things where we'll start taking energy from them are pretty much the heart and the brain. And when we've been fed, then we have the opposite effect start to happen. So when we've been fed, then we have levels of glucose that will start to rise, and this actually triggers our pancreas to stop secreting the glucagon and start making insulin. And so this is where we start to have kind of an interaction between the signals from our food and the signals coming out from our body. And what this allows us to do is to start storing away all of that glucose into glycogen for long-term usage. And then if we have other components of the food, if we're eating something that has a protein component and has a lot of amino acids, then we're going to use those and begin storing them as fat and start using that for later. So there's a multi-step process here, but usually these things are going on in parallel. So the next question here is then, how do we take all of these different nutrients that we are getting and trying to store long and short term and get signals to the brain so that we can change our behavior, decide whether to eat or not? So there are a few different signals that happen, but one of them that is found to be very important is a hormone that's called ghrelin. And so ghrelin actually is secreted by the digestive system, but it also acts like a neuropeptide when it gets into the brain. And this is one kind of neurotransmitter. So ghrelin is released directly by the stomach, and as well as parts of the intestines when they are empty. And so when we are in a stage of fasting, we have really high levels of these, and you can see this here. So if you look at the point where an individual has breakfast, then at this point, then the levels of ghrelin in plasma or in blood are much, much higher. And then as soon as the meal is finished, then after a little while, the ghrelin levels drop. And then just before a meal, they start rising again. And so you can actually predict when somebody had eaten pretty much by this pattern of ghrelin in their body. And when ghrelin gets passed through the bloodstream, it's going to go up to the hypothalamus, which we're going to see a lot. Many parts of the hypothalamus, just like we saw with thirst, are very close to the bloodstream, and they have components where they can get information that passes either across the blood-brain barrier, or just like when we looked at the lamina terminalis, we will find areas of the hypothalamus that are receiving these signals that fall outside the blood-brain barrier, because it is their job to interact with what's going on in the bloodstream. Another thing that we know is that if we directly inject ghrelin, then we can make people think that it's time to eat. So if we artificially increase levels of ghrelin, then you actually end up with a perception, even though there's no physiological need, you end up with a perception that it's time to have more food. Another way that we get these signals to the brain are through detectors that are out in the periphery. And so we know that we have certain detectors that are in the liver that can detect glucose, as well as kind of the lipid content going through the blood. And so when we have a deprivation of these, then that'll trigger feelings of hunger. And one set of detectors, as I said, are found in the liver, but the other set are found in the brain. So we have some other glucose detectors that are up in the medulla, which is actually back here in the brainstem, and not where they have that labeled at all. They're just showing that generically as the brain. And so the signals get to the brain in a couple of different ways. Of course, the ones that are in the medulla are having direct contact, and we think that the ones from the liver are coming up through, likely through the vagus nerve, which carries a lot of information from kind of the intestinal system up to the brain. So that gives us some idea of how we might detect that we're hungry if we have these glucose or lipid receptors that are telling us that we don't have enough of something in the system. But we also have to have signals that say that we've had enough, or that we're satiated, that we're full. So the brain can get a number of different forms of satiety signals. And one of the things that I want to remind you of is that if we're thinking about these correctional mechanisms, we usually have signals that are short term that are telling us, you've had enough to eat, your stomach is full, but they're not actually, those are really monitoring how much of a correction we've made. And then eventually we have feedback that comes all the way back in and says, okay, no, you have enough energy in your system. But it takes a long time for us to actually measure whether we truly have enough of all of the right nutrients. So we have short-term kind of corrections that we work with, and then we have a much longer process as well. So the short-term signals, their main job is to stop a meal, and they're really signaling things happening just really mostly in a few minutes, maybe in a couple of hours, but mostly over a few minutes. One of the most effective signals that the brain has is actually kind of its own predictions. So over time, we learn to predict things. And so by having just some memory that we've recently eaten, or the understanding that we've recently eaten, we use this as information. And we also have brain systems that are involved in predicting what the meal size was. So you all know about how much you think that you can eat in any given time. And this is where the phrase, oh, my eyes were bigger than my stomach comes from. Sometimes this is true. Sometimes the amount you predict that will be enough to fill up your stomach is far more than what you actually needed to eat, and your stomach starts to send signals prematurely. So part of what goes on in this prediction is it's not just a volume thing, but we actually predict how certain foods will make us feel over a long period of time. And this is where we get into trouble sometimes, because we all really, really love these foods that are high fat and high sugar, because those are the two that give those great signals back to the brain that say, hey, this is really good, you've given me really good nutrients. And this is where receptors in the entire digestive system start to give us some of that feedback. But our bodies over time have evolved to want to detect sugars and fats, and we don't have as robust of a system for detecting proteins or other things like that. And so the things that become really kind of large driving forces, or what we might say salient, they have the most value to them, are these fat and sugar signals. Another signal that comes in the short term is that we have levels of Ghrelin to decrease, and so that's the one we just saw on the graph a minute ago, but coincident with that, we have levels of two other feeding peptides increase, and one of them is colchicystokinin, or CCK as we usually call it, and the other one is peptide YY, or sometimes called neuropeptide Y. And so these are going to increase when we're satiated, and then they are going to decrease when we start to get hungry. So they kind of act as negative feedback signals. One of the other really important short-term signals is insulin. So insulin, of course, is being used to make all of these conversions between glucose and glycogen, so that we can take all these sugars and we can store them for kind of short to medium term usage. But something that makes a whole lot of sense for building the entire body and brain together as a system is that the brain actually has insulin receptors. So when we have these big increases in insulin, then they start to tell the brain that we're nearing the point of satiety, and we should listen to those signals as well. In the long term, we actually use signals from fat cells. So much like we saw these cells in the thirst system that can expand and contract, as our fat cells expand, then we're able to detect the size of them. And this is actually the main thing that determines body fat percentage. So these cells can get bigger and bigger. And when we have fat cells that are doing really, really well, they produce a peptide called leptin. And this basically tells our brain that we've had enough to eat because our fat cells are happy. And this acts as a long-term satiety signal, although it's not a perfect satiety signal. And it could, because of course, one of the things you're probably sitting there thinking as you're sitting at home, probably watching all of your classes and getting to snack on whatever you want, is, well, then why don't we just give people leptin to get them to stop eating? And there were a number of studies that tried this, and it turns out that it doesn't quite work. So it's not it's sufficient to signal that we're getting close to satiety, but it's not necessary to stop feeding. So there are multiple interacting features here. And so if you give people leptin, they do start to report things like feeling more full, but at the same time, it's not a perfect way of stopping feeding. We need multiple sets of signals working together. Now we're going to move right into part 3. If you haven't taken a break already, this might be a good point to do so. There are a few more slides left to get through, and it's definitely worth putting your full attention into this section. What we're going to be talking about here is how all of these interactions we've been looking at between brain and body with regards to thirst and hunger can sometimes fall out of balance. And what that means is that, among other things, we're going to be talking about eating disorders. So I know that for some individuals this might be a trigger, and so I'd like to just take a moment here and give you this trigger warning. You'll know what's best for you in order to approach this material, whether that's looking at the textbook or waiting for the appropriate time to go through this last section of the video. But in case this might act as some kind of a trigger for you, this is your warning that the slides that follow are going to contain that kind of material. So what we've seen already is that many aspects of food intake and also liquid intake are controlled in parts of the brainstem. We've seen both pathways interact with parts of the medulla, including the nucleus of the solitary tract and also the area postrema. These are the places we also saw in the gustatory system that we're getting input from the tongue. And this also happens to be the same area that's getting some of the signals we talked about from the intestines and the stomach. Beyond this, it is the area of the brain that contains detectors for glucose and is what signals the brain to start taking action when glucose levels fall. So what we see is that if we go and use electrophysiological recordings and record these neurons, then you have parts of the medulla that show more action potentials when animals are hungry, and you can also activate this area using electrical stimulation, and you can start to signal the body to cause it to start pulling nutrients from long-term reserves. So it seems that there's a lot of information passing through, especially the nucleus of the solitary tract, which is part of the medulla, that gets the information from the body and then can also pass information back to the brain to tell it what behaviors we should be engaging in or what things that the body should be doing to maintain homeostasis. And it turns out that if you lesion this area, and this is, remember, we use this for loss of function, that we can prevent feeding when we have signals from the body that should be telling us it's time to feed, that you eliminate the nucleus of the solitary tract, and now those signals no longer matter and they no longer cause the initiation of feeding. We've also talked a lot about the hypothalamus, and we looked mainly at the lateral hypothalamus, but there are actually two areas of the hypothalamus that seem to be important: the ventromedial hypothalamus and the lateral hypothalamus. And these two are the yin and yang of feeding behavior. So if we lesion the lateral hypothalamus, we stop eating or drinking, and we've already talked about this 4 or 5 times. This is the area that it was discovered that if you stimulate this area, you cause animals to voraciously start eating and sometimes drinking, depending on where you stimulate. On the other hand, the ventromedial hypothalamus does exactly the opposite. So if you lesion the ventromedial hypothalamus, you start to cause overeating, and electrical stimulation of it will suppress feeding altogether. So the lateral hypothalamus seems to be an area whose functional role is to generate feeding, and the ventromedial hypothalamus seems to have a role of suppressing feeding or a role in signaling satiety. One of the types of neurons in the lateral hypothalamus that seems to be involved in this are a set of neurons called melanin concentrating hormones, or MCH hormones, and another set are orexin neurons. So both of these neurons have been shown to have roles in stimulating hunger. And we're going to keep coming back to this, but this diagram, we'll come back to it a couple of times, shows all of the other signals in the brain starting with these two neurons in the lateral hypothalamus. So when we think of lateral hypothalamus, we really start to think feeding center. And remember, we're getting information from just across the blood-brain barrier to these neurons in the hypothalamus in many cases. Another set of neurons that are found in multiple parts of the hypothalamus are these, we already talked a little bit about them, but we said peptide YY neurons before, but they're also known as neuropeptide Y. The only difference is where they are. So we tend to talk about peptide YY when it's released in the body. And then when it gets into the brain, we now start to call it neuropeptide Y. And these neurons seem to be important for stimulating hunger. So they're found in a part of the hypothalamus that is kind of right near the middle of the brain and right next to part of the ventricles, these fluid-carrying passages of the brain that also can take a lot of information in from cerebrospinal fluid and from the body and other places if it's chemical information. So these neuropeptide Y neurons are found in this arcuate nucleus. And when animals are food deprived, we see that these neuropeptide Y levels go up and they decrease following a meal. So these, remember, we have the same signaling coming from the body. We already talked about that in part 2. So the brain sees this same change in levels of the neuropeptide as we have changes in peptide YY in the body. And if we infuse neuropeptide Y directly into the hypothalamus, we can drive eating behavior. So this suggests that this has a very critical role in hunger and food consumption. So what's been done with a couple of more recent experiments is that you can take viral vectors, which are basically viruses that are kind of like this coronavirus going around. They can bind to things, but these viral vectors have been engineered so that they have some other mRNA or DNA inside of them that encodes for something that we want to use as a tool. And most of them, unlike our current coronavirus situation, they don't replicate. They can't infect people, and this is why we call them viral vectors. They're just viruses used for the delivery of genetic material, and that's their only job, and then they stop once they've done this. So a lot of experiments use this kind of approach now in order to be able to either upregulate, to increase, or to downregulate or decrease levels of a certain mRNA or protein in the body. And what we found is that if we increase the levels of neuropeptide Y by injecting a viral vector that causes the cells in the arcuate nucleus to produce more neuropeptide Y because now they have more copies of it in their genetic sequence, then this will actually increase food intake. And then if we do something that's opposite of that and we insert or use a genetic targeting to knock out the current copies of that gene that's already there, then it can actually decrease food intake and counteract even obesity in mouse models that are obese. So the neuropeptide Y neurons seem to get a few different kinds of signal, but we already talked about there's this peptide YY signaling from the body, and it also seems that they have receptors for Ghrelin, which was that, remember we saw that really pretty graph in part 2, where Ghrelin levels increase right up to the point of a meal, and then they decrease afterwards. So there's some synergy there between. Another interesting thing about Ghrelin is that it has interactions with parts of the brain that we think of as being reward centers or reward systems. And so there are receptors for Ghrelin on the dopaminergic neurons that are in a part of the brain called the ventral tegmental area that is thought to be critical for reward processing. But it's really critical for a number of different things. And this is kind of the debate about what these neurons do. One of the things that they may do is sort of process reward, but in a way that relates to some sort of drive or effort state or motivation to obtain certain rewards. And so if you think about it in that way, it might make sense that these quote unquote pleasure neurons have Ghrelin receptors and that they can be activated by Ghrelin. The interesting thing, though, is that if you infuse Ghrelin and activate these neurons, then it actually starts to stimulate feeding. So somehow there's a bit of a feedback loop here where activating the reward system in this precise way with Ghrelin not only causes the feeling that something is rewarding or starts to initiate effort, but it leads to a specific kind of effort that seems to drive feeding behavior. And in probably one of the more timely slides for us to talk about, given some recent New Jersey propositions for the election, cannabinoids are another source of signaling that can change hunger. And when we talk about cannabinoids, we're not talking about marijuana and the external signal, although that does affect this system. What you may or may not know is that there's an internal system in the brain where we have a set of neurotransmitters that we call cannabinoids. And of course, they're named based off of cannabis. That was one of the major things that we first found to mimic them. So these molecules are actually at their highest during fasting, which is something interesting to think about, right? Because whenever cannabinoids are high naturally, that's at a point when we're hungry, and they're lower during feeding. So what happens if somebody does smoke marijuana, for example, then? Well, that stimulates these high signals and can then trigger eating behavior, which is kind of the very well-known stereotype that goes along with marijuana. And the opposite of this is enzymatic degradation, which is, of course, the term you should remember is one of the ways that we get rid of neurotransmitters by breaking them down. So if we disrupt the breakdown of endocannabinoids, it causes weight gain and obesity because it leads to kind of a long-lasting signal of hunger when one does not necessarily exist. And on the other end, we know from a more valuable, I guess, clinical application that cannabinoids can be used clinically to increase appetite in certain patient populations. And so this is something that's often used in, for example, in cancer patients. So at least for certain clinical utility, especially when you compare to the use of opioids, cannabinoids have some role in decreasing pain, which is interesting. And it's not really something that we've spent much time on, but also in increasing hunger. And so, especially for populations where those are two major problems, and as I said, cancer patients is one of those, there can be some really valuable clinical utility. The New Jersey proposition is an entirely different bag of worms coming from a substance abuse researcher by myself, like myself. So that's maybe something we can get into when we start to talk about psychopharmacology. We'll leave that for today. So in thinking about all of these signals, there's a really good thing to know here is just sort of a general overview. And I think that things in part 1 and 2 about just what the signals are very important, and then putting that all together with what you have here. And so some of what we've talked about is actually really well summarized in this diagram from your book. So I would take a look at that, and you can kind of go back and understand just the role that especially each of these different neurotransmitters like the MCH or melanin concentrating hormone, orexin, neuropeptide Y, and so on and so forth are all playing in this bigger system of feeding. And for these last few slides, we're now finally going to get into our discussion of eating disorders. I'm going to go through these kind of quickly because I think that there is some common knowledge information, and so we're just going to focus on some of the details that are important for really what's preceded this of how signals in the brain and body interact and might contribute to various specific eating disorders. So if you're really interested in a lot more information, I do think the book has some more detail, and as always, you're welcome to dive in a little bit deeper. We'll go through these pretty quickly, and then that'll be it for the day. So factors for obesity in the United States and around the world currently is that one of the main things is we just have an incredible amount of high calorie foods that are available. Fast food is everywhere, highly processed foods. And one of the things that is in many of those foods is fructose. So this is a form of sugar. But one of the interesting things about fructose is that it doesn't actually activate the body in the same way as glucose does. And so by not activating the body in the exact same way that glucose does, it creates some problems for what the body is trying to do. And we no longer signal hunger in the exact same way as we might have normally. Another thing that's happened is, of course, we started out maybe as a bunch of hunter-gatherers or something like that, where we had to work really, really hard to get our food. But now we've gotten to the point where physical activity is at an all-time low, and many of these signals that come from the body are influenced by exercise. And so not only are these signals out of whack because we're putting new chemicals like fructose into our bodies, but we also have reductions in physical activity because we sit at desks or in classrooms all day or whatever have you. And then the last of the major factors for obesity is, of course, genetic factors. And one of these that was found very early is that some people have a deficiency in leptin production. And so those individuals that have deficiencies in leptin tend to be obese. And this specific genetic disorder accounts for very, very few people in the world. But it did lead to a number of studies on leptin. And when leptin was first discovered, researchers hoped that this kind of naturally occurring neuropeptide could be used to treat obesity. And in fact, there was one drug company that paid a really large amount of money for the rights to develop a compound for this. But what it turns out is that most people that are obese have really high blood levels of leptin, and if you increase this with injections, then it actually ends up having very little effect. And so the system is already out of whack, and that's why leptin therapies tend not to work, or at least not the prior versions of them. Who knows, maybe there will be future genetic manipulations that can reduce all of this. The main treatments for obesity come all the way back to these different signaling cascades that we've talked about. And so one of the most common are bariatric surgeries. So the bariatric surgeries do a couple of different things. The first is that they reduce the amount of food that can be taken in at one time. And in some cases, they even reduce the amount of kind of stomach and intestinal pathway that food can pass through, which means that you actually have reduced absorption of food as well. And the two most common of these are the gastric band and gastric bypass. And my understanding of these has always been that they play off of the fact that you remember we use receptors in the stomach that stretch out when we have a lot of food there as one satiety signal. And so we make that satiety signal happen more early. For obesity treatments, anything pharmacological has typically had poor outcomes, and we just talked about the leptin story. That's a perfect example. So there are very, very few things that work, even though there are a lot of promises in this world. If you sprinkle this packet over your food, it's magically going to make you thinner. If you drink this skinny mocha Java coffee that your friend on Facebook is selling to everybody, then you're going to lose 30 pounds in 10 days. And these things all tend to be quite overplayed because we have a lot of data out there that they don't really work very well. And then the last thing is that we have behavioral interventions, and these really tend to have the strongest effects, and they're most often used ahead of bariatric surgeries, and in cases, because remember, we do have genetic components here, and we also have neurochemical imbalances and all kinds of other things. So there are some individuals who need a bariatric surgery or some sort of corrective action to take place. But for others, behavioral interventions tend to be incredibly worthwhile. And so if you can get people to start exercising and change, kind of their behaviors along the way, then those kind of behavioral outcomes tend to be the very, very best, because they then cause people to have lower blood pressure and increased bone density and all these kind of things that come with the benefits of exercise, as well as producing the reduction in obesity. So the second thing we're going to talk about here are eating disorders. And there are a few major eating disorders. We're not going to spend too much time on this. I think that you're all fairly well aware of what these eating disorders are, but the main categories are anorexia nervosa, bulimia nervosa, and binge eating disorder. And for all of these, there's a greater prevalence in women than there is in men. So one of the things that happens in both anorexia nervosa and bulimia that makes them kind of common, they have something in common with each other, is that there is a purging component to both of these. And so in the case of anorexia, you tend to mainly have restricted eating and just a fear of gaining weight whatsoever. And this can lead to a lot of different problems because the body is just starving all the time. And so this is sort of almost a compulsive attempt to prevent weight gain. And then in bulimia nervosa, you have episodes of binge eating that are followed by periods of purging behaviors. And so this ends up being kind of more of a yo-yo. And then in binge eating disorder, there tend to be large binging episodes, but there's none of these kind of compensatory behaviors like the various forms of purging that come along with this. One of the things that we know happens to the brain in the case of anorexia is there can actually be a shrinkage in brain tissue. And if you think back to what we talked about in the case of hunger signals, this begins to make a little bit of sense. So the main reason that we think that this happens is because starvation first draws from glucose and then draws from fat tissue and takes the sugars and the triglycerides and lipids that are in our fatty tissue. And then the third thing that happens is we start to eat away at proteins. And so in the case of anorexia, we actually start to see degradation of the brain that may be partially due to an inability to produce the right number of proteins, or potentially even, in fact, the almost the consumption of our own bodily tissues. So one interesting thing about eating disorders is that they don't appear to link to any other mental illnesses. And it's taken us a long time to figure this out because there are so many mental illnesses that have comorbidities where there's some other illness that comes along with them. But instead, this seems to be just some problem with our ability to regulate our food intake and other things. And part of the reason it was so difficult to detect this is because we do see a lot of symptoms that match other disorders like mood swings and depression. but these can all be linked back to starvation behaviors. So an interesting thing about starvation is that we actually know that this increases levels of physical activity. And so as you get to a point where you're hungry, then you'll start to move around a lot more, and animals do this. In fact, animals in a laboratory, if they're given restricted food, they tend to live longer and be much more active than mice or rats that have free access to food. And we've seen that this over and over again relates back to that neuropeptide Y circulation. Another interesting factor for eating disorders that is worth knowing is that of course, there's genetic factors here, but there's also biological trauma that can contribute to this. And in the case of many of these eating disorders, this biological trauma comes about as sort of a self-perpetuating process. So by continuing to engage in fasting or purging behaviors, what ends up happening is we cause further damage or imbalance to the brain and the body, and then these signals become even more amplified, and then they make the disorder worse. And this is the case with many mental illnesses or mental disorders, whichever terminology you prefer for that. But this is something that comes about because of the extreme behaviors that happen there. So in wrapping up, one last factor that we've mentioned a little bit but not talked about in depth is that there are sex differences that contribute to eating disorders. And we've eliminated some other factors like comorbid disorders as a part of this. So what that means is that there's a true sex difference, something based on the biology. And there's a lot of good data supporting this, but your book shows just one example. And what they look at is women who come into the lab, and in the first week they're not food deprived, and so they'll eat some amount of food, and as it turns out, if you're not food deprived, men will eat more. That makes a lot of sense. But something interesting happens when a week later you ask people to come back, and they're now in a fasting state. So what men do is they eat more food during the laboratory study to compensate for the fact that they're hungry. But when women are fasting, they actually come in and they eat less. So fasting triggers some sort of reduced eating state, and this suggests that there's an inherent sex difference in the way that things are handling, and it's possible very quickly for lack of food intake to drive an even greater lack of food intake in women. And this could very easily contribute to something like anorexia, which tends to be the main eating disorder that is more common in women than men, and likely relates back to some major biological difference.